So it is a great privilege to be able to introduce Eddie DeRobertis, who most of us know as a really wonderful colleague, and a generous collaborator, and an absolutely spectacular mentor to his students and postdocs. He's also an investigator with the Howard Hughes Medical Institute, a fellow of the American Academy of Arts and Sciences, and in 2013 was inducted into the National Academy of Sciences. He has many other national and international awards that are too numerous to actually mention. But for the last few decades, Eddie's research has focused on elucidating the molecular mechanisms underlying the establishment of the vertebrate body axis. And his contributions have been immense. In fact, I'm in the process of teaching developmental biology right now, and in the textbook I use, I would say that over half of the material in 3 of the 15 chapters comes just from Eddie's lab. In 1985, Eddie was a professor at the University of Basel, and he cloned the first vertebrate Hox gene there in collaboration with Walter Gehring. That was the first vertebrate gene known to control development. And with that event, that cloning, the field of evolutionary developmental biology was born, now known more widely as Evo-Devo. And more recently, Eddie's been investigating the molecular mechanisms that establish the dorsal ventral axis of the vertebrate embryo. And that work led to the, at the time, completely unexpected discovery that the cells, the organizer that control the axes, actually produce BMP antagonists. And this sets up a gradient of BMP activity across the embryo. And as with his work on Hox genes, this BMP gradient has now been shown to control DV axis formation in many bilaterian animals. So today, Eddie's going to tell us about the recent work from his laboratory and looking at interactions among the many signaling pathways he's discovered in the organizer. And so we'll learn about the integration of FGF, Wnt and TGF-beta / BMP, morphogen gradients in development and cancer. So Eddie.
Thank you very much, Karen, for your kind introduction, and thank you for the invitation to Mark and Deborah Krakow for participating in this seminar where I have been so many times as a listener. I would like to discuss with you today morphogen gradients during development and a little bit of cancer. And I will divide my talk into 3 parts. First, I will tell you that the BMP antagonist cordon forms a morphogen gradient in a very narrow extracellular matrix highway that separates the ectoderm from the endomesoderm. This is the first time that we can actually see an extracellular morphogen gradient in a vertebrate. Second, I will tell you that Wnt signaling stabilizes many proteins that are phosphorylated by GSK3. And finally, how one of these proteins, tumor suppressor SMAD4, also called deleted in pancreatic carcinoma, serves to integrate the TGF-beta, nodal, FGF, and Wnt pathways. And this is interesting because this was a protein that was thought to be an unregulated component of the TGF-beta signaling pathway, and we now find that it is a regulated one. And the frog embryo is a perfect tool to study, oops, okay. Mark, help. Okay, this was a movie. Okay, there's a movie of an embryo dividing, which doesn't seem that it's going to, oh yeah. And so the embryo divides from 2 cells, then to 4, and to 8 and 16, and so on. And so this is the sperm entry point here. And in the frog embryo, what you see is opposite to the sperm entry point, there's a region of less pigmentation. That is going to become the future dorsal side, and it's marked by a rotation of the cortex of the embryo. And this region is going to have a Wnt signal. And these cells at this point, they're only signaling to each other. They're not differentiating into anything. So that's why it's so good to study signaling here. At this stage already, the zygotic genes are turned on, and there's gradients of nodal, gradients of FGF that are instructing these cells. And now we're going to have a look from underneath this embryo, and you're going to see that there are cells that move to the inside of the embryo. So this is underneath. Look at this cell and this cell. You're going to see they migrate into the inside of the embryo, where they will form the endoderm and the mesoderm. And these movements take place once in your lifetime, and they're called the gastrulation movements. And at the end of this enormous morphogenetic activity, you end up with a body axis that has a perfect head to tail axis, head to tail, and a dorso-ventral axis. So you would think that that's extremely difficult to obtain. And in fact, it's even more interesting that if you, let me see if I can, something with the movies. Okay, this movie was going to show that we can take an embryo and cut it in half.
You may have to do the movies on your computer.
Yeah, well, okay, in here.
Just put the button on it.
Well, it doesn't matter, doesn't matter. If you take this embryo and you cut it in half, and you take the dorsal half and the ventral half, it will rescale, so it will form a fairly good half embryo if you have the dorsal part. If you don't have it, the ventral side, you will just get no axis and just ventral tissues. But if you take great care and you cut the dorsal to ventral, and so that it has both dorsal organizer and ventral organizer, what you find is that you regenerate the perfect embryo. So this is the ultimate case of regeneration. You regenerate half the body. So where you would have half, you have the total embryo. And the way to study this, which would seem an impossible task, was shown by an experiment done by Spemann and Mangold in 1924. And what they did is they took a little that fragment where the cells were moving, and they transplanted it into the ventral side of an embryo. And then from that, then you get a twinned embryo that forms a perfect Siamese twin. And they showed that the transplanted tissue had this power to induce new cell differentiation in the host. So that was a new, for example, a new brain, new somites, a new eye. And so that was the discovery of embryonic induction, for which Spemann got the Nobel Prize in 1935. And now we know that this experiment is explained by the production of inhibitors in the dorsal side of the embryo, such as cordin and noggin, which was discovered by Richard Harland at Berkeley. And there is a gradient of BMP activity to the ventral side. And from this gradient of BMP activity, that's what determines the different tissues in the vertebrate. So you have on the ectoderm, you have at low BMP, you have central nervous system. At high BMP, you have epidermis. In the mesoderm, at low BMP, you have notochord. Then as you go higher, you have muscle. Then you have kidney. Then you have lateral plate. And then you have blood. So all of these tissues come from a gradient of BMP, and similarly in the endoderm, which would be this region here. So the question is there one gradient or many gradients? Are there 3 gradients or even more gradients of BMP? And so that's the question that we're going to address today. And the gradient can be visualized by looking at a transcription factor called phosphosmad158. And you see in the nuclei on the ventral side, you have much more BMP signaling than on the dorsal side. So that's an indirect measure of the gradient. And so you see less on the dorsal than on the ventral. And we've studied this over the years. and found that it would be enough to have a lot of cordon, say here, to get a dorsal embryo. But as we studied that, we found that there are not only dorsal components, there are a lot of ventral components. The whole embryo participates in this. And in fact, there are dorsal BMPs, which we predicted had to bind to cordon and then flow to the ventral side in a flux, and then where they would be liberated by an enzyme, extracellular, this is all extracellular, by an enzyme that it will degrade cordon and release fresh BMP on the ventral side. So we did that all based on embryological experiments, but we had never been able to see this. And now we can see it because we have improved immunostaining methods, so we can see the gradient of cordon. And it was found in a very interesting region, which is this region called Brachet's cleft, but it's just the space, the extracellular matrix, that separates the ectoderm from the endomesoderm. And this is present in all embryos, not just the frog embryo. And so you have the ectoderm, and you have the mesoderm and the endoderm. And you have to imagine this extracellular matrix region extends all the way around the embryo, from the dorsal all the way to the ventral side. And it is here where the cordon gradient is established. And therefore, here you could, cells moving along this, probably as they're moving along, can sense this gradient both in the ectoderm and in the mesoderm. So it would suffice to have one gradient of BMP antagonist or of BMP to get the patterning of at least these 2 germ layers or the 3 germ layers. And the way it looks, it's like this. So with a cordon antibody, you see this is the distribution of the cordon protein. And if you put a morpholino, the staining goes away. So if you knock down cordon, it goes away. And if you compare this expression to the expression of the RNA is much more localized to the dorsal, while the protein is expressed in the dorsal, but also it diffuses quite a way along this region of the separation between the 2 germ layers, the ectoderm and the mesoderm. And if you look on the sagittal section, you see RNA is only expressed on the dorsal side, not on the ventral. But if you can see that it actually can reach even the ventralmost Brachet cleft. And so this, you can see that it extends over the entire embryo. So you can scan this by densitometry all the way this way. You can see you get a peak going up and then going down. And what I wanted you to note that the distance here of this quite smooth gradient is very long. This is 2 millimeters, because this is a 1.3 millimeter egg, which has over 10,000 cells. So this is, the whole embryo is participating in this. And this extracellular matrix is very rich in fibronectin, which is what provides that. And you see the fibronectin is uniform, but the cordon is not. And it extends all the way to the ventral. So if you have this gradient, what would happen when you do this organizer experiment in which you put a little bit of tissue from the dorsal lip, you transplant it to the ventral side, and it forms a perfect double embryo. So somehow there's a field here that is divided into 2 fields. So you would expect a new gradient. And in fact, this is a graft with membrane-bound GFP. And so this is now staining for cordon. You see this is where the graft is, and this is cordon extending outside of the graft. This is another graft. You have this one, transplant, and cordon moves outside of the graft as well as from the endogenous organizer. So when you put a dorsal lip, you get 2 gradients. And then what about this half embryo experiment? When you cut the embryo, then if you look at the phosphosmat1 gradient of BMP activity, the dorsal half regenerates the BMP activity, and so you have the gradient back. And on the ventral half, since there is no opposition to the BMP, there's no cordon, no noggin, it goes on and on signaling and reaches levels of signaling that are much higher than the you would find in the whole embryo. And when you look at the expression of cordon, you see that on the dorsal half embryo, you regenerate a cordon gradient. You see it here in the brachet cleft. You don't see it on the ventral side. This is the ventral half, no cordon, so this would be the background. So this is the cordon gradient. And this then, cordon is a very abundant protein. If it were uniform, you would secrete 30 nanomolar of cordon in extracellular space. But now you can, in that entire embryo, but if you now think this is now concentrated greatly in this very narrow extracellular matrix. So where it's reaching high concentrations, and there'll be cordon and BMPs, and that's what is establishing the gradient. And so cells can probably sense this as they're moving along, like in that movie that fortunately I could get working, and then they could be reading this gradient as they move along it in the extracellular matrix, which we consider now like a signaling highway for this tissue. And then it can pattern both the ectoderm and the mesoderm in one go, because the cells come into contact with that and are probably reading their position information from that. So for people that like embryos, this is a new kind of vision of this signaling highway, and there's some precedence for that. So that's what I wanted to tell you about cordon, which is this stage of the embryo. So that's, you get the organizer, and that patterns the BMP gradient. But there are earlier signals that trigger where the organizer is going to be formed. And these are first a maternal signal that you saw in that pale region of the embryo, where you will have a Wnt signal, so right from the egg, right from the very beginning. And then from zygotic transcription, you have an FGF gradient that you can measure by a gradient of diphospho-ERK, which can very efficiently stain embryos. And you have a gradient of nodal, which are a TGF-beta family member, which form a gradient mostly in the dorsal. That's what we call the Newcoop center in our jargon. But so there are three gradients that will come together into this. So our interest in how this might work came from some work that we did on the Wnt signaling pathway. And what we found is that when Wnt signals, GSK3, which is an enzyme, becomes localized to multivesicular bodies. And let me explain how this happens. This is a cell it's a 3T3 cell transfected with GSK3-RFP. So this is a cytoplasmic enzyme. It's one of the main kinases in the cell. And it's more or less uniform, we would say. But now if you drop a cell near it expressing Wnt venous, so that would be the Wnt signal, the untransfected mouse cell is going to pick up Wnt, which from the neighboring cell, and into these little vesicles, which are endosomes. But now look what happened to the GSK3. The GSK3, instead of being uniform, now moves and rushes into the endosome. So it goes and tries to follow the Wnt into the endosomes. And so we've been able to work this mechanism out. And what happens is when Wnt binds, it activates a certain receptor. And GSK3 is in a destruction complex together with axin, destruction complex by which beta-catenin gets phosphorylated and degraded. When you get Wnt signaling, the receptor complex is endocytosed. And people thought, well, this is what must be signaling. But we realized that when an endocytosed activated receptor, their usual destiny is to go into the lysosome. And for that, there's an obligatory pathway through the multivesicular endosomes. So you must go through these interanuminal vesicles. And when that happens, then GSK3 and axin become sequestered from the cytoplasm. So then there is less GSK3, it goes into this, and then you can accumulate proteins such as beta-catenin that won't get degraded, and then it can go into the nucleus and have its effects on transcription. So from this, we would say that GSK3 sequestration causes protein stabilization by decreasing cytosolic phosphorylations that are normally required for protein degradations. Proteins have what are called phosphodegrons. So when you have very close by phosphates, then they're recognized by E3 ligases that then degrade them. And so more than an inhibition of GSK3, it's a sequestration. But GSK3 has many, many substrates. Are they affected by Wnt too? And so people knew already by when we were doing this work, there were at least 40 others GSK3 substrates. So GSK3 means glycogen synthase kinase 3. So what we did is to screen the human proteome for proteins having 3 or more GSK3 sites. So GSK3 is an enzyme that prefers pre-phosphorylated sites. It has a priming site, and so if it binds to a phosphorylate priming site, if 4 amino acids upstream there's a serine or threonine, it phosphorylates it. And then with the same enzyme in a processive way, then puts this one in the priming site, and then phosphorylates that one, and then searches more, phosphorylates that one. And an incredible 20% of the human genome contained 3 or more of these GSK3 sites. And that's much more than you expect by chance alone. Of course, it's a lot of proteins, but maybe you have a protein you like, and you might want to know if could it be regulated by Wnt. You can go to our web page, and there you have something called GSK3 proteome. So it's a list of proteins, and you can type in your protein, and it'll tell you if it has these sites. 20%, so that's a lot. Some of them are well-known proteins, like beta-catenin. You expect that. SMAD1, which we had shown, is phosphorylated by GSK3 and regulated by Wnt. The one that came out that's the most conserved is this family, MITF, that is studied by Diego Plopper in our lab, and he's found that it is regulated by Wnt, so this is predictive. And then what I want to tell you today is about this one called SMAD4, because SMAD4 is a very important tumor suppressor gene, and it is also part of the TGF-beta signaling pathway. So we knew that BMP can be regulated by Wnt, but for TGF-beta, this is not known if there is any crosstalk between TGF-beta and Wnt. And in fact, there are not good GSK3 sites on SMAD2 and 3, so we thought, well, maybe these phosphorylation sites and these threonines present in SMAD4 might bring in regulation to this TGF-beta pathway through Wnt, through this pathway. And this is now the work of a graduate student that just graduated in France, but he devoted his thesis to try to work out this, and his name is Hadrien Demagny. And the first thing that Demagny did was to make an antibody against 2 of these phosphosites in SMAD4. It turns out if you can have 2 sites, you get very high titer antibodies. So it's very effective for a phosphospecific antibody. And so this is a Western blot from just plain fibroblasts, L cells. You see they contain endogenous SMAD4. There's an excellent monoclonal antibody for that. But they don't have very much of the phosphorylated form of this protein. But if you add FGF2, then it gets phosphorylated. And if you pre-incubate with Wnt, then you inhibit the phosphorylation. So that would be this step here. Wnt will inhibit these phosphorylations. And if you put an inhibitor of GSK3 called bio, you will completely block the phosphorylation, or block it quite a lot. So in order to find out how many phosphorylation sites there are in this protein when you add FGF, he used 3T3 cells and this monoclonal antibody. You see, if you have no serum, there's a single band. But if you add FGF, you get 1, 2, 3, 4 phosphorylations. He used a trick, which is called the Phostag gel, with which many of you will be familiar. It's just a chelating agent that is put in the acrylamide gel, and then the phosphates bind to this and they get retarded so that you get a good separation. And these 3 bands on the top are in fact mediated by GSK3, because you can see that they are stained by the phosphosmad GSK3 antibody. And if you now, and these are indeed GSK3 phosphorylations, because there's an inhibitor called lithium chloride, and when you put lithium chloride, then these 3 bands go away, and you have just a single band which will correspond to the MAP kinase. As you will see later, this is the most active form of SMAD4, the monophosphorylated form. It's biologically active. And if you then block the ERK pathway, so you have FGF and something that blocks the ERK pathway, U126, you completely block everything. So even if you have FGF, you don't get, so the priming is required. And we can confirm that the priming is required by making phosphoresistant mutations in this. And you have, for example, if you knock out the MAP kinase site, or if you have the GSK3 mutant, in which you put, instead of threonines, you put alanines or valines in this, you will then lose all phosphorylation by the antibody when you add FGF. And so that's what that is. I think I'm going to need another pointer. If you had another pointer, that would be a very good thing. And so this has consequences, then, this phosphorylation, which are that SMAD4 is stabilized by the Wnt RNA. This can be evident in this experiment, in which we do it in the animal cap, so that's a very efficient system, because if we take dissociated animal caps, the MAP kinase, thank you very much, is activated very greatly. And so here we took embryos, injected them with flag SMAD4 mRNA, and it makes this much protein by the start of gastrulation. But if you add a little bit of Wnt mRNA, the protein is stabilized, and this is after mRNA injection, so it's stabilization of the translational step. But if you add the mutant form, then Wnt has no longer any effect. So that tells you that this is going through the GSK3 sites that we have described in this. So the way this works was worked out by Hadrien de Magny, and he found that when you have all 4 phosphates, there is an E3 ligase called beta TRCP. It's one of the most common ones, and it was known to bind to SMAD4, but it was not known was that this was regulated by phosphorylations. In fact, it was not known that SMAD4 is ever phosphorylated. It was thought to be a constitutive protein. So you can see here, these are plain 3T3 cells in which he immunoprecipitated SMAD4, and a little bit of beta TRCP, stained with an antibody, is pulled down. But if you now put FGF, which phosphorylates all 4, you get much more beta TRCP. And if you put Wnt, which inhibits these, then beta TRCP doesn't bind. So this is then a regulated phosphodegron by these proteases that put the polyubiquitin. But you will see that it's not just the degradation, which we can reveal in the frog very well. In cells, that takes several hours, but the effects of FGF and Wnt are much quicker. So it cannot be just, to see the degradation, we can see it, but you have to put cycloheximide and so on. So we think that there is also a transcriptional activation domain, activity in this linker region. This is a transcription factor, which I'll show you in a minute. So the key question now is Wnt regulating TGF-beta. So for that, you use a reporter gene for TGF-beta. It's called CAG12, and it's a standard TGF-beta reporter. So you have minus TGF-beta plus TGF-beta 80-fold. So it's a nice induction. Now, if you put Wnt and you put minus and plus TGF-beta, there's absolutely no difference. So this is as people thought. These are two separate pathways. One is the Wnt pathway, the other one's the TGF-beta pathway. So they're insulated from each other. But look what happens when you add FGF. FGF will phosphorylate all of these, so this activity is a little bit lower. But now, when you add Wnt, it goes up. In fact, it goes up even more than in the TGF-beta alone. So we were thinking of finding a regulation between Wnt and TGF-beta, but we found that these are all linked. It's like cog wheels. Like you need the FGF, and if you have the FGF, only if you have the FGF, the TGF-beta signaling is stimulated by Wnt. So that's the thing. And this is going through the SMAD4 because there are many cancer cell lines in which have mutated SMAD4, because SMAD4 is a major tumor suppressor. And so this is a mammary cell gland, mammary tumor. And so if you add TGF-beta to it, of course, there's no signaling because there's no SMAD4. If you put TGF-beta and cells have been transfected as SMAD4 wild type, you get TGF-beta signaling. But now you add Wnt3a to the medium, it greatly stimulates. So then this effect requires SMAD4 and if you now use the SMAD4 GM mutant, so it's this here, you find that the signaling is high, but it lost completely the response to Wnt. So that's telling you that the response to Wnt is going through these particular sites. So the crosstalk between TGF-beta and Wnt is mediated by the SMAD4 GSK3 phosphorylation sites. And remember that I told you that this is higher than in the wild type. And what happens is that the MAP kinase monophosphorylation is a positive step in this signaling. And we can reveal this, for example, in experiments that we can do with the frog. That's why frogs are very good to study signaling. If you put a little bit of beta-catenin mRNA, you get a partial twinned embryo. But this requires SMAD4, so you put SMAD4 morpholino, no twinned embryo. If you replace it with SMAD4 wild type, you get the twinned embryo back, so that's as you expect, because you put SMAD4 morpholino. This is human protein, messenger RNA. But now if you mutate the GSK3 sites, you get even stronger axes. So these are shown here, so that's the GSK3 mutant. But now you mutate one more residue, and you lose the activity, so it's this here. And but now, if you just replace one more amino acid, you put this one into a phosphomimetic aspartic acid, then you get back the activity, and in fact, it's the most active form. So what this tells you is that MAP kinase phosphorylations activates the SMAD4 signaling. And in fact, the SMAD4 linker is enough to have this activation. Then you can show that by taking the DNA binding domain from GAL4 and putting just the linker part of this protein, just 140 amino acids here. And when you put FGF, then you get this much signaling. But if you put FGF and Wnt, no, you can restore the signaling. So the signaling is all working at this transcriptional activation domain level. And if you put the GSK3 mutants, then you lose the difference between these two. And there's no great difference in the amount of protein. So this is not, the protein stability is at the transcriptional activity intrinsic to this transcription factor, which is then, it's a growth factor regulated transcriptional activator. So what's going on is that when you have Wnt, if you have FGF, the MAP kinase goes on, and this gives you a more active protein. Presumably it binds a co-activator, and it's been described that this, more or less this region of SMAD4 binds P300, so that's probably the likely co-activator, although we haven't studied that. But once you get the phosphor GSK3, this cannot happen anymore, and so this now is inhibited. And we imagine later, because we see a delay of a couple of hours, then you bind the beta TRCP and the protein can go and be degraded in the proteasome. So what we think are two stages: first the inhibition of the activation, and then the beta TRCP of this protein. And this crosstalk, sometimes it doesn't look like that much. For example, here, if you put 100 nanograms of TGF-beta and FGF, you must always have FGF, it's only 1.5 times minus Wnt or plus Wnt. So it doesn't look like that much. But this is extremely concentration dependent. So if you take the same data and you plot it as indicated down here, you see that to get 240-fold stimulation of the TGF-beta reporter, you need 100 nanograms per ml of TGF-beta. But if you have Wnt, then this becomes much more sensitive, and it's enough to just have 1 nanogram per ml of TGF-beta. So it's a very big difference at low TGF-beta concentrations. That's what we imagine happens in tissues in vivo, that there would be low levels of this TGF-beta signaling. And so, we think that it produces a very big effect. And then you might ask, well, what about the embryo? What happens in the embryo? In the embryo, the organizer genes, such as cordon, expressed here, are turned on by two signals. One is a maternal Wnt signal that goes through beta-catenin, siamois, and then the organizer genes. And then there's a second signal called nodal, which is a TGF-beta, which causes phosphorylation of phosphosmad3, and together with SMAD1, SMAD4 turns on the organizer genes. So when you put SMAD4 morpholino, is here, so this will be inhibited, then you don't get organizer activity. But if you put SMAD4 wild type from humans, then you rescue the formation of the organizer. But if you put the SMAD4 GSK3 mutant, you get much more organizer. So we think, from this experiment, that these sites are very important in limiting the amount of organizer that you get through these phosphorylations of GSK3, which are also mediated through this Wnt signal. Now, this also gave us an entry to understand a very puzzling effect of something called competence modifier. 20 years ago, Doug Melton and Randy Moon found that the animal cap, when you inject Wnt, it doesn't induce mesoderm. However, the injection of Wnt, particularly in dissociated animal caps, enormously increases the response to activin, which is activin induces mesoderm. It's a TGF-beta. So they call that competence modifier effect of Wnt. So what we did is to make a reporter gene for this part of the pathway that is not sensitive to this one. It's a reporter gene from the cordon promoter. And if you take these animal cap cells and dissociate them like they did and have them plus or minus activin protein, if you put SMAD4 morpholino, since you don't have SMAD4, you don't get any response. But now, if you put the SMAD4 wild type, you get response to activin. But if you put Wnt to it, then you get even more response. And if you put the mutant, the SMAD4 GSK3 mutations, then you don't get the response to Wnt anymore. So what this means is that Wnt increases mesoderm induction by TGF-beta through SMAD4, in this case, in the Xenopus embryo. So part of the competence modifier effect is going through this mechanism of SMAD4, which seems to be very sensitive to this integration of signaling pathways. And this is seen even better when you look at a marker for mesoderm. There's this band here represents the RNA for a gene called brachyury, which is a pan-mesodermal marker in Xenopus. And so if you put the SMAD4 morpholino, which it's a mixture of 3 morpholinos that were developed by Stefano Piccolo, a previous student of ours, if these morpholinos will completely block brachyury expression. Now, if you put the SMAD4 wild type, this is recovered to a surprisingly normal-looking embryo. So you get that. So very good rescue, as you would expect, because you now have wild type SMAD4. But if you put the SMAD4 that's been mutated in the GSK3 sites, now you get the entire upper part of the embryo turned into mesoderm. So what that means is that there is no stopping the mesoderm induction, and then you get no ectoderm. You just get all mesoderm. So this tells you that this phosphorylation at this level is very important for the specification of germ layers in the embryo. So that's a very striking effect. So as these things go, that's a very strong effect of these poor phosphorylation sites that we just found by digging in this collection of genes. So then, so that we think that it's very important, this particular regulation so the GSK3 normally is stopping SMAD4 in the animal cap from turning everything into mesoderm. And so in the embryo, what happens and why this is interesting to us is you have this intersection of 3 signaling gradients. One, the maternal Wnt signaling. You have this zygotic FGF signaling that you can see with the diphosphoric antibody, and then the vegetal pole nodal signaling. And on the dorsal side, you will get a perfect storm of all 3 come together to give you the maximal amount possible, according to this scheme, of SMAD4 activity and of TGF-beta activity. You remember that in the presence of FGF, you can turn on TGF-beta activity with much lower levels of signaling, even in the presence of Wnt. So all 3 come together. So for us, that's important because this sets up then the Spemann organizer, the expression of cordon, and that will give you the gradient of BMP in which we started today. Now, these things have a little bit to do with cancer also, because SMAD4, like I said, is a very major tumor suppressor, and many, many metastatic cancers have deletions in SMAD4, also called deleted in pancreatic carcinoma. Although this runs in families, actually the mutation in SMAD4 can be deletions or point mutations, is a very late event in cancer progression. Most cancers start by activating the FGF or EGF or RAS pathway. And that stimulates proliferating genes such as cyclin D and c-MYC and many others to make the cell proliferate. Another pathway that is frequently activated early on in cancers, and many times they're both together in cancers, is the Wnt pathway, which can be activated by loss-of-function mutations, for example, in adenopolyposis coli or in axin. So these are loss-of-function mutations that give you an activated Wnt, and they also stimulate the proliferation of cells. But now look what happens on the other side of this. If you now have activated RAS, you will get a lot of phospho-ERK. And of course, in the absence of Wnt, this will decrease the activity of SMAD4 or be neutral, because it'll get phosphorylated by GSK3. But if you have also Wnt, which is very common to have RAS and Wnt in the same tumor, what you're going to do, you're going to increase the signaling ability of SMAD4. So you'll have an activated form of SMAD4. And this activated form of SMAD4 can then cooperate if there is TGF-beta, and we assume most tissues have some bit of TGF-beta, and then you will have the phospho-SMAD3 together with the very active SMAD4 will help on the anti-proliferation effects of TGF-beta. And so this, by turning on cycling D kinase inhibitors, CDK inhibitors, such as P14 and P21, which are known targets of TGF-beta signaling. So TGF-beta has anti-proliferative effects, but when you have these two, you will increase the anti-proliferative effects, and it will balance the proliferative effects. So it's a game of balancing. But then when you lose SMAD4, it's a completely catastrophic effect, because this was linking 3 signaling pathways in one protein. And in fact, this is many tumors have deletions when metastatic of SMAD4. And it's more than any other parts of the pathway frequently. Now, you can also have deletions of the TGF-beta receptor or SMAD2, but by far the one weak link in all this is SMAD4. And we propose that it's the weak link because it serves to integrate 3 signaling pathways. And remember, this is a protein that was previously thought to be unregulated by any growth factor. And so, well, that brings me to the end. And so I should thank the frog embryo and thank my collaborators, Hadrian Demagny and his undergraduate Tetsuya Araki, and for the cordon part, Jean-Louis Prunek, Yuki Murayama, and Lise Sakin. And I will leave you with these thoughts that there is a BMP gradient that forms in the extracellular matrix, a signaling highway between the ectoderm and the mesoderm, that when signaling stabilizes many proteins, and that SMAD4 phosphorylations integrate the crosstalk with FGF, Wnt, TGF-beta, and I didn't tell you, of course, also with BMP, which is part of this. So thank you for your attention.